Welcome to another screencast where I'll be talking about the pre-lab and the lab itself for bacterial transformation. And so let's just get started. So the first thing we're going to do is in the incubator back here, which is set at 37 degrees C. You see in the incubator here, these are the plates, right, where our E. coli are growing. Right? E is the genus name for Escherichia, and then coli is the species name. And just a little pet peeve here, this gets underlined when you handwrite it. When you type it, it gets italicized. It's the scientific name for these bacteria. So they're growing on those plates in the back of the room in the incubator. So you're going to go back there. You're going to get a plate that has some E. coli growing on it. Now, a little anatomy of the plate or the petri dish here. There is a top. This is the top. I don't know why that's not writing. That's the top, and this is the bottom. And the top is larger than the bottom, so the top fits over the bottom. And in the bottom, this is the growth medium, or the agar. Right? So this is sometimes also known as the nutrient agar. And in this nutrient agar, there is a broth called LB broth, not Lake Braddock, just fortunate that it has that same initial. And in there, in that broth, there are sugars, and there are amino acids, right? Stuff that the E. coli are going to eat, grow on, right? This is the nutrient agar, so the nutrients in there are stuff that the E. coli cells are going to metabolize, and it helps them live. The agar is gelatinous, and I'll pass samples of that around in class, and so you can see and feel what that's like. So we pour the plates, we pour the agar in the bottom of the plate, and then the top of the plate sits over top of it. But we store the plates upside down, like in this picture here, so that just in case you pick up the whole plate, you would grab the top there and you'd pick it up and you didn't open it. Because if it was sitting like this and you grab that, the problem is then you open this up and the plate comes, the lid comes off of there, and then it's exposed to the air. So as it is right now, this is sterile. So as soon as you open up the lid, what's happening is all the little bacteria and things that are in the air are now going to fall onto the plate and will now start growing on there, and then your plate is contaminated. So you don't want that to happen, so you store them upside down thusly. That said, when I was pouring them this time, we had a little bit of contamination. So you'll see some of the plates, some of these starter plates that look like this, and this is where the contamination is in the red X right over here. So when you go to get your E. coli, do not grab bacteria from here. Right? These are E. coli right here, right? And these are colonies of E. coli. Right? So each little individual one, there's a colony, that's a colony, that's a colony. Right? So this is what a non-contaminated starter plate is going to look like, and then here it is large. Right? So if you're looking at this from the side, right, here would be the bottom of the dish. Here's the nutrient agar, and then the E. coli are growing, the colonies are growing on the surface of that agar. Okay? So there's a little bit about that. Moving on, some other tools you're going to use. These are micro centrifuge tubes, and so you have the relative size to these things, so they're small. They have a lid that will close, and here it is open. So we have microcentrifuge tubes we're going to be using. These are the microcentrifuge tubes. Some of them are loaded with a sample of LB broth, and then others of them are labeled with TS for transformation solution. So the transformation solution is a calcium chloride solution. So in the lab, when you're reading over, that's what this looks like. So each group's going to get some microcentrifuge tubes with LB broth and transformation solution in them, and then this tube is going to be empty, and this tube will be empty. Also, other tools you're going to use. This is an inoculation loop. This is an inoculation loop. You can see it's a loop because here we go on the end right there. So this is, they're going to come sterile in sterilized packages like this. So this is sterile. You're going to open it, break the seal here, open this up, and pull one of them out. So now this is where you're going to grab this end with your hand. This end is still sterile, right? But as soon as you take that out, it becomes non-sterile. But you want to use it as quickly as you can, right? So this inoculation loop is going to be used to grab E. coli colonies from the starter plate and transfer them to the microcentrifuge tubes. Right? And these loops will also be used when transferring the plasmid, right, as well to the samples. So there's inoculation loops. And then you have sterile pipettes. Right? And so these pipettes are graduated, and there's a line here. This is the line for 1 milliliter. Right? This is the one halfway down is 500 microliters or 0.5 ml. So that's 500 microliters. This is 1,000 microliters, same difference. Right? This line is then the 250 microliter line. Right? This would be a 200 microliter. This would be and then that would be a 100 microliter line. Okay? So these come sterile as well. In the packages, you'll open up the package, pull the bulb out a little bit here to grab it with, right? And then do your transferring. Okay? So again, you're going to use microcentrifuge tubes, sterile pipettes, and then inoculating loops in the lab here. What are you going to use them for? Well, here we go. One of the first things you're going to do is you're going to use one of these pipettes to move transformation solution into each of the tubes. Right? And then you're going to use another one of these pipettes, strike that, to move nothing. Sorry, I goofed up there. But you are going to move transformation solution into each one of those tubes right there. Right? You're also going to label these, the tops of these tubes here. We'll label this one plus and label this one minus. Right? So plus and minus on the tubes. Next thing you're going to do is you're going to move colonies of E. coli into each of those. Right? So again, this one was labeled plus, this one is labeled minus. You're going to use a separate inoculating loop for each one. So inoculating loop 1, you're going to come and you're going to scrape off a colony. Right? Don't dig down into the agar, just scrape the colony off on the end of that inoculating loop, and then that gets transferred into this tube. Then you're going to use, so that's 1, then you're going to use another inoculating loop, go up and grab another colony off your starter plate and put that into the other tube. Right? So again, 2 separate inoculating loops, 2 separate transfers of E. coli into there. Right? Then you're going to use another inoculating loop, right, to come back and get a sample of the plasmid. And the plasmid is in this little vial here, and it gives you the relative size, so there's not a lot. And then you're going to take this inoculating loop, and you need to transfer 10 microliters of, this is a volume, of this plasmid into this plus tube. Right? So the 10 microliters is the volume that would fit across this here. So if you've ever blown bubbles, you know how you get the bubble solution in it, and it makes a film across the loop there. Same thing here. You're going to put this end into the plasmid sample, make a little bubble, make a little film across there like you're going to blow bubbles, and then that plasmid is going to get transferred into the tube that has the plus. This plus indicates that you are adding plasmid. So plus, right, means that you added plasmid to your sample. Right? After that, you're going to take that whole float there, that whole, all these microcentrifuge tubes. Here it is from the top view. Here it is from the side view. And you see that those tubes stick out the bottom of this styrofoam float thing right here. You're going to put that whole thing into a 42 degrees C hot water bath for, I believe it's 50 seconds. I have to go back and look, but I believe it's 50 seconds. And then after that, it goes onto back into the ice. Right? So this is the heat shocking part of the lab, and then after you do that, and after you heat shock them, you're going to use another sterile pipette. Right? And again, this is the plus and this is the minus. You're going to take this sterile pipette and transfer LB broth into each of these tubes here. This is after the heat shocking. Then there's some incubation time, setup, etc. And then finally, you're going to take samples from, whoops, this is not a plus. This is minus. Take samples from each of these tubes and then apply them to new plates, right? So you have a plate that has just LB broth in it. You have a plate that has LB broth in the agar as well as the antibiotic ampicillin. You have another plate that has LB broth and ampicillin. Then you have a plate that has LB broth, ampicillin, and then arabinose added. So the labels on the side tell you what's in each plate. Right? So this is going to be an LB. This is the LB minus plate. This is, you're going to have to mark this on here. This is going to be the LB amp minus. This is going to be LB amp plus, and this is going to be LB amp minus. Don't mark on the bottom. You're marking on the bottom of the plates, but don't make like a huge mark, you know, on the plate like this, because then you won't be able to see through it. Okay, so don't do that. You are then going to take a new pipette, a sterile pipette, grab some of the, this is going to be plus, sorry, LB amp arrow plus. You're going to take a sterile pipette and take a volume from here, spread it onto this plate. You're going to take then another one, take plus and put it onto this plate. Take a sterile pipette, grab some of this minus, put it onto this plate. Take another sterile pipette, grab some of this minus, and put it onto this plate. Right? After you do that, you spread that volume all around using an inoculating loop or not. I forget exactly what the protocol calls for, but we'll look that up before we go. And then after this, you're going to take all your plates, stack them on top of one another, and you'll put them into the incubator, again, at 37 degrees C. Right? And you will then check these plates after about 48 hours. Right? And we're going to see if we get any growth or what the growth looks like. Okay? So hopefully that was helpful. This will help you sort of step through that pre-lab. And if you have questions, bring them to class. Bring them to class and just be ready to go. We get kind of one shot at this, so there you go. Thanks for listening.